First of all, thank you, Jennifer, and thank you everyone for organizing this conference. It's been super exciting set of talks and very thought-provoking and inspiring. And I apologize for not being able to troubleshoot in the last days. The airlines mess has been a joke. Yeah. Okay, so yeah, first of all, a little bit of background about my lab. So we are interested in understanding the mechanisms of learning and memory in general from a system neuroscience perspective. And to do that, we have a multidisciplinary approach. So we use electrophysiology mainly. And when I say electrophysiology, I should clarify that is extracellular recordings. We use closed loop manipulations and cell type specific manipulations to get to the circuit. And then we also have a variety of behaviors, mainly in the lab, well-controlled behaviors to control for the learning. But recently we have also started to be interested in how all of these processes expand to more close to natural behaviors. And so today I will be focusing on some of the recent data that we have collected in the past year in my lab that I just started related to the consolidation phase of memory. And of course, I think Yasunori just made a better introduction for this, but just is a key brain region for memory in the brain is the hippocampus. We know that since Brenda Milner and colleagues discovered that removal of the hippocampus impairs the abilities to form new memories. So we always look at the hippocampus at least as part of the regions of the brain that are important for memory. And from a broader, more system neuroscience perspective, the current view of why the hippocampus is involved in memory can be summarized first with the finding of place cells that have been described by several of the speakers already. Place cells are just pyramidal cells in the hippocampus that fires in specific locations as an animal is moving through an environment, such as if the animal starts in the home port and move towards a reward location like here, then a set of place cells will cover the whole trajectory. So that will be like a meaningful representation for the animal. And more recently, this has been extended to reward cells, to identity cells, and so we can more generally talk about receptive fields and ensembles of cells that represent anything that is relevant for an animal during exploration, active behavior, and encoding phase. And then after this learning phase, there is the so-called consolidation phase, which involves, as Christine was just mentioning, Sarway ripples. And Sarway ripples are oscillations of 150 Hz that one can see in the hippocampus. And it has been shown that during this oscillation, the cells fire in the same order that they did during behavior. So that has been a very strong suggestion that they are also involved in memory. And this has been reproduced by many groups now throughout the years. And furthermore, manipulation experiments have been applied to prove that selective disruption of Sarway ripples during behavior or during sleep impairs memory. And more recently, the generation of artificial Sarway ripples, this is something that can be done with a smooth pulse application of light to hippocampal pyramidal cells or interneurons, can also be used to improve memory. So in putting together these two steps, the learning phase and the consolidation phase, this summarized what it has been known for years as the two-step model of memory, which is that there is a first learning phase where the animal picks things from the environment that are relevant for behaving or for engaging in that behavior. And then there is a second phase or consolidation phase during which these same cells are reactivated in a compressed window of time during Sarway ripples. And there are many things that we still don't know about the mechanisms of how this happened, both in the encoding phase and in the consolidation learning in pre-non-REM and after the learning in post-non-REM. So this means that the animals are sleeping before the learning and sleeping after the learning, and the cells are measured during Sarwi ripples in those stages of the sleep before and after. There is an increase in the firing rate of the cells that have been recruited during the behavior. So this is shown here in the red trace. And in parallel, and this might be related partially to this finding, but in parallel, the fact that they are recruited together during Sarwi ripple also increase the correlation between these cells. So if we look at the cells before the learning and after the learning during this specific, let's say, privileged windows of time, the cross correlation between the neurons that are firing together is also increased. So these neurons have become an ensemble that fire together after learning. And so this is easy to understand and to explain why Sarwi ripples can be involved in memory, but something that we have been worried a lot in the past months is that this supposed, because both things implicate an increase in firing rates and correlations, this also is introducing an imbalance in the network, because if assuming that the network wants to be within a certain range of balance, this is moving the firing rates and the correlation of cells up towards increasing, and assuming that animals have sometimes very heavy memories or very rich environments to explore, we can take things to an extreme just to motivate the question, and the firing rates cannot keep increasing, and the synchrony cannot keep increasing upon a threshold, because then things will go pathological, and this is something that is characteristic of many diseases in which the hippocampus is key as well. So the question that we set up to investigate is like, how does the hippocampus compensate for these very precise changes that is able to do in order to support memory? And so in order to do that, we have, this is the schema of our recording. So we have bilateral electrodes in the two hippocampus, and we do that so we can cover all subregions of the hippocampus. In case someone is not familiar, the hippocampus has four main subregions, CA1, CA2, CA3, and the dental gyrus. So we want to look at everything. And for this talk, I will be talking about CA1 and CA2 mainly. And then we are using several tasks. This is the first one that we started using. So the animal is first in the home cage during a baseline sleep, so to use as a control. Then we expose the animal to two novel objects that have to explore during several trials. Then the animal goes back to sleep during the same amount of time than before. And then there is a test where we have moved one of the objects, and so the animals, this has been shown in the literature, that the animal explored the novel object, suggesting that it remembers that the location is different. And so to start with the controls to make things that we are doing things right, we of course see Sarwi ripples during the sleep. So this is how the Sarwi ripples look like in the CA1 and in the CA2 region. There is a spiking. There is an LFP trace at 150 Hz. And if we looked at the rate of Sarwi ripples, so the incidence of how many ripples we get per second in the post-sleep stage, we can normalize that to baseline, assuming that there is like a pre-existing rate that Sarwi ripples happen. There is an increase at the immediate consolidation phase, and that it is decreased with time. So this has been shown in the literature many times, so we should see this effect as well. So we are happy, we are doing things right, but just to make sure that we were taking the results to a robust stage, we tested the animals in several other memory tasks. One is the T-maze alternation task. So in this task, we are using it because we think it's a little bit more demanding for the animal, because the animal has to wait for 5 seconds and then move toward the center stem and left, right, left, right. And we also have like sleep before and after. And we also tested the animals in a different task of some sort of different nature, which is a social memory task. In this case, the animals have to explore two novel mice that they haven't met before. And we swap the position of these mice to make sure that it's not space dependent. And then for the test, in this case, we introduce a novel mouse, and the subjects explore more time the novel mouse, suggesting that they remember the previous mouse. And we again see Sarwi ripples in these tasks as well during the post-sleep, and we plot the rate of ripples, and we see an increase of incidence of ripples in the post-sleep in all of the tasks that we have tested. So that is good because it has been shown previously in the literature. But we followed closely. Is there any question? So we followed closely the consolidation period, and we were looking carefully at the hippocampal network during this time. And well, first, yeah, this is the single cell responses to Sarwi ripples. So far, this is how the LFP looks. It's 150 oscillation. This is how we identify the Sarwi ripples. And then we look at the single cell. So here we have a raster of 3,000 cells that we have recorded, organized per subregion. So we have the CA1, and then we have the CA2 cells separated per group because we have previously identified that they show an heterogeneous response to Sarwi ripples with a subgroup of cells responding earlier and then becoming quickly silenced after Sarwi ripples. And then another subgroup being highly active, propagating the activity towards CA1. So this indicates there is a network effect. Most of the regions are entrained and recruited during Sarwi ripples. And then we followed closely the consolidation phase, and we found that in addition to Sarwi ripples from the LFP or raw traces of the recording, we can identify other periods of time that look like this in the picture. So this is an increase, a massive barrage of action potentials from CA2 pyramidal cells that can last for almost a second. And so we followed these periods and the rate of incidence of these events, and we saw that they also increased during the post-sleep, very much like Sarwi ripples, the same trend. So at the beginning, they are the highest, and then they decrease in time. And we have tested this in the different tasks, and we see consistently an increase of, we are calling this CA2 barrages. We see an increase at the beginning and then decay with time along the consolidation phase. So next we look at the cells. We have the same raster, so the cells are organized in the same order here compared to Sarwi ripples. And what we see is that those cells that are quickly silenced during Sarwi ripples, now they are very highly active in long periods of time, almost half a second. And some of the other CA2 cells are also active, not as much, but also active. And the surprising thing is that the CA1 cells are not responding very much. We can also, once we have identified these two events, we can just get a timing and then we can do a cross-correlation in time between these two events. And we found that when there is one of these events, CA2 barrages, there is almost not a Sarwi ripple until 3 seconds after, suggesting that this is a one or another kind of situation. So the network is either producing a Sarwi ripple or producing a barrage within a second's time scale. The next thing that we looked is LFPs. So LFPs are like the population activity. We can measure what are they doing, all of the cells together. So during Sarwi ripples, there is the very characteristic 150 Hz oscillation. So this is a frequency spectrum triggered at Sarwi ripples in the CA1 region and in the CA2 region. So the 150 Hz is similar in the two regions. And at the bottom, I'm showing a frequency spectrum during CA2 barrages in the CA2 region in the right and in the CA1 region in the left. And what this shows is that during barrages, there is no oscillatory component. There is not so much synchronous. It's rather a broad spectrum increase in the higher frequencies, suggesting that it's just spiking during long periods of time and a pretty much silent situation in CA1. This can be quantified just with the peak at 150 Hz for ripples here in black for the two regions, and just the broad increase in the CA2 region in the higher frequencies representing the spiking. So just to summarize the characteristic features of these events that we have identified during non-REM sleep, They happen anti-correlated with ripples in time. They recruit a subpopulation of the CA2 region of the hippocampus that are very active during what we call CA2 barrages, and they are quickly silenced during ripples. And the CA1 region seems to be mostly silenced or not very active. And this is what we set up to do next. So this is a weird network state where the CA1, which is a region that is the output of the hippocampus, is very important. We all look at what are the CA1 cells doing during behavior and during memory. So why there is this network state where the CA1 is not very active, and what are the cells doing? So what we did first is to separate the cells in those that increase the firing rate during the task, during the tree of the task here in the 3 different colors. And this is just a separation based on firing rate. So we compare the increase in firing rate during the behavior compared to baseline, and we separate those that are mostly increased during the task versus every other cell. And we see here that the cells that fire or have increased more the firing rate during the task are more recruited during Sarwi ripples after, and compared to the others, they are preferentially more recruited. And if we look at this CA2 barrages network state, what we see now is the opposite. So the cells in the CA1 region, the cells that have been more increasing the firing rate during the behavior are less active during CA2 barrages compared to the others. And this was just the first pass looking at firing rates because it's easier. But then, of course, the next thing is to look at places, to look at the cells that are functionally, or I'm quoting functionally, but like more related to the representations that the behavior of the animal indicates. And this is example place cells for the different tasks. So we identify place cells versus what we are calling non-place cells. And we see that the place cells are more recruited during Sarwi ripple as well in the post-sleep, but they are less recruited during CA2 barrages, in line with the previous results based on firing rates. The next thing that we did is to look at ensemble of cells together, because the single cells activity represents unique location in the space or unique features of the space, but presumably what is important for the animal is an ensemble of cells that represent something entirely meaningful, like a full trajectory or an identity of an animal in the case of the social memory task. So we apply a first method to detect ensembles of cells. This is called assembly detection method, which uses first PCA and then ICA to identify cells that are coactive together. So we do that, we detect during the behavior the ensembles of cells that are coactive together, and then we follow those ensembles of cells in the post-sleep, and what we see is that they are positively, compared to pre-sleep, recruited during Sarwi ripples, but they are very much inhibited during CA2 barrages. Just to make sure that the results were robust, again, we apply another method. This is called explain variance, which detects pairwise correlation of cells during the sleep, in the post-sleep stage, in relation to the learning stage or encoding phase, subtracting the pre-existing correlation between these pairs of cells. So this is just multivariate analysis to remove the baseline activity in terms of correlation between cells. And what we see again is the same thing, that the cells are the ensembles of cells that are more coactive together are more recruited during Sarwi ripples in the post-sleep and less during CA2 barrages. So this was sort of to compare and follow the cells that are important for the behavior of the animal and look at the post-sleep in these new network states that we identified, but we were also interested in comparing, because Sarwi ripples happen also during the baseline. So we followed now the CA2 barrages stage to the pre-sleep, and we look at the cells in the 2 sessions of the sleep. So we follow the cells in the post-sleep, and we compare to the baseline pre-sleep session. So we have here in this plot, I'm showing how much a given cell increased the firing rate in the post-sleep compared to what was already firing in the pre-sleep during Sarwi ripples in the y-axis and during CA2 barrages in the x-axis. So we can call it firing rate gain during specific events. And what we see here is that these two events show an anti-correlation in terms of the firing rate gain or change of the cells from the pre to the post-sleep session. So the more a given cell increase the firing rate in the post-sleep Sarwi ripple, the more that cell gets down activated or inhibited during CA2 barrages in the post-sleep. And this is something because this is the pool data. So this is something that we can again see for all of the tasks. So it seems like a general stage of the hippocampus that is independent of the task and is in the post-sleep session present no matter what the animal is doing. As a control, because it has been proposed that the sleep can decrease the firing rates over time, is one of the main hypotheses of the sleep at the single cell level. So as a control, what we did is to better understand the role of this CA2 barrages state, we plot the gain of the firing rate of the cell during CA2 barrages here in the x-axis, and we took shuffle or random periods of time during the same sleep stage. So this is the non-REM stage, and this is the number of events of the shuffles that we are taking is the same number of barrages to control for the number of events. Is the duration is similar between 500 milliseconds and 1 second. So everything is as alike as possible to these events. And what we see is that the baseline firing rate of the cells is correlated during random periods of non-REM and CA2 barrages, suggesting that it's only the relation with Sarwi ripples which is anti-correlated. And of course, we wanted to understand a little bit more of what is behind this. So we cannot go all the way to the molecular mechanisms that some of you have discussed in the previous talks, but we can look at the cells and we can, in particular, look at the interneurons because for Sarwi ripples, there is a lot of literature suggesting that PB fast spiking interneurons are responsible for pacing the frequency of the cells at 150 Hertz. So this has been shown in spontaneous Sarwi ripples. In red, this is the firing of pyramidal cells. In blue, this is the firing of interneurons. So these interneurons can follow such fast frequency, and it can be even induced artificially. And if someone can apply a smooth light pulse to the hippocampus, it can entrain a ripple oscillation. So it seems like it's a unique property of the PBs to be able to pace the cells at this frequency. So we looked at the interneurons then during these CA2 barrages, and what we found is that there was a superpopulation of cells that was increasing the firing rate as well during these interneurons, during these two CA2 barrages, while another superpopulation was very much not changing the firing rate. So this is shown here in the box plots. And now we keep these groups separated as they are, and then we looked during Sarwi ripples. And what we saw is that now the group reverse. So the interneurons that were not changing the firing rate during CA2 barrages are very much recruited during Sarwi ripples. They can even follow the fast oscillatory component of the 150 Hertz, and the interneurons that were slightly more active during CA2 barrages were less active during Sarwi ripples. So again, both at the pyramidal cell groups and in the interneuron subgroups, there is this dichotomy between cells that are recruited by one event or another one. We have compared the waveforms and the autocorrelograms of these subgroups of interneurons to verify that they are indeed different. So we are just trying to see if we can get the features that can identify or can genetically differentiate between these two subgroups of interneurons. And to do that, or to get more idea of what to look for, what we did is to calculate the cross-correlograms between pairs of cells. So this is, we get the spiking train of one pyramidal cell and we get the spiking train of an interneuron, and we cross-correlate it, such as we can see whether or not they have a peak over the threshold of the baseline within the monosynaptic window which operates in the hippocampus, which is between 3 to 5 milliseconds. So this is one of the interneurons that were active during Sarwell Ripple, so presumably parvo albumin positive interneuron. And when we do this monosynaptic cross-correlation, what we see is that these interneurons receive most of the inputs from cells that are really close to that particular interneuron. And when we do the same for the interneurons that are active during the CA2 barrages, what we see is that they receive most of the inputs from pyramidal cells that are farther away, or compared to the other, these inputs are coming from farther away distances. And now these interneurons, by the way, are both CA1 located interneurons. So we finally can quantify all of this, and what we saw is that the interneurons that are active during Sarwell ripples receive most inputs from CA1 pyramidal cells, so matching the local distances, and the subgroups of interneurons that are active during CA2 barrages receive most inputs from the CA2 pyramidal neurons. So as a summary, what I have shown you today is that there are alternative network patterns in the hippocampus during the consolidation, or what we have called the consolidation phase. In particular, here today I show you the CA2 barrages that involve a subgroup of CA2 pyramidal cells that are really active for 500 milliseconds all the way up to 1 second, during which the CA1 region instead is mostly silent. And both of these events, Sarwell ripples and barrages, recruit different subgroups of interneurons in a way such as the CA1 pyramidal cells that are preferentially reactivated during Sarwell ripples after learning are also more silent during CA2 barrages after that learning. And so to close up with the same schema that I started, this is the model that we all use to look at the system level for reactivation, replay, and consolidation. So there is the encoding phase where the place cells are active. There is the consolidation phase where the reactivation happens during Sarwell ripples of the same cells. And now what we think is that the model can be updated to take into account network events during which the hippocampus balance precisely the changes in the firing rate of the cells that have encoded that particular memory. And this is supported by internal hippocampal circuits that recruit one or another event and target the same subpopulation of CA1 cells. So this to end the talk, what I will say is the same statement with which Leslie started her talk, which is that I think there are more and more evidences to think about this two stages model of memory rather than in one phase under the other. Multiple processes are happening in parallel at a fast time scale with an envelope of a slower time scale that might be modulating all of it, all the way down to molecular mechanisms. And so this is Lindsay, a PhD student in the lab, the one that mainly developed experiments in this story, Ryan Harvey and Heath Robinson, who helped with analysis. And I would like to thank my funding sources and the members of my lab.
Okay, thank you very much indeed. So we have time for some questions.
Hi, thank you. Super interesting. What terminates the barrages? How long are they?
How long are they? That's the easier part. Is between half a second to 1 second. What terminates them is a really important question that I don't have the answer for yet. But Well, presumably it's the same as in Sarwi ripples. So there must be a interplay between excitability. The I balance at the beginning will take over in the C2 region at the beginning of the barrages, and then at the end, inhibition will take over. But I don't know the detailed mechanisms of how that might end.
Yeah, the CA2 cells have interesting ion channel properties because the PV interneuron activation didn't seem to track the full extent of the barrage, right? That was like a couple hundred milliseconds, not a full half second or second. That's related to my second question, which is CA2 is pretty small, and this is a subpopulation of CA2 pyramidal cells. How do you think they exert such a powerful effect on CA1? Is it through those PV interneurons?
I think is so the CA2 is pretty small. That is true, but it has unique properties. And to me, the role of CA2 within network processes that makes it unique is the ability to follow both a very fast network activity through the higher density of PV that they contain, and very slow processes through the connection with neuromodulatory centers that they have. Mm-hmm. And I think that it's a powerful way to change what I call the operation mode of the network in the hippocampus. So you can trigger recurrency, you can pull inhibition. It's very versatile region for moving the network one way or another.
Thank you.
Let me ask a question. So do we know, what do we know about the place cell, about the place properties, for instance, of the CA2 cells? So do they have spatial cell activity as well?
Yes, that's correct. They have both place cells and non-place cells. They do not have the same trend as the CA1 cells during CA2 barrages, if that's your question. So the anti-correlation, the very precise anti-correlation with Sarwi ripples is not maintained in the CA2 region. So I think in this case, there might be other properties that the CA2 has, but in this case, the CA2 is acting more as a modulator or triggering the network state for the CA1.
You don't see sequences. There's nothing like the spatial sequences you see in CA1 during these
barrages. No. Oh, yeah. I see. No, we don't see that. No, we have those controls to see whether these are, one can think that these are special ripple oscillations that happen locally. It's not that. We don't see replay, we don't see reactivation. Everything is non-significant for correlations during this event. So it's more of an asynchronous state within the local network of CA2.
I'm sorry if I missed this, but where do you think CA2 is getting, what input's driving this?
That is another very important question, which I don't know yet. We have some preliminary evidences that this might be facilitated by a high cholinergic tone locally in CA2. But other than that, it could be entorhinal. Entorhinal has a powerful effect in CA2. It could be, maybe this was related also to Rob's question before, oxytocinergic modulation of CA2, which also has a powerful effect for controlling the output towards CA1, but we don't know yet. My best bet so far, because of preliminary evidences that we have, is cholinergic.
Are there different sets of cholinergic inputs, or the receptors are different in CA2?
Not that I know of. There are inputs from the supramammillary, which is a strong cholinergic modulator. There are inputs from the medial septum and lateral septum. Receptors-wise, I don't know. My guess is more like it's a subset of neurons that are receiving the particular inputs.
Thanks. Thanks. These barrages are very, very long, and because of some of the plasticity mechanisms that we've been hearing about, I was wondering, do cells that fire during the barrage, do they fire throughout, or do they take turns?
In CA1 or in CA2?
In CA2.
The cells that fire throughout the barrages are very few cells, but they fire throughout. So the C2 cells can produce we are calling it like super bursts, because they are like bursts that are, compared to CA1 and CA3, super long and with a high number of spikes. So for some reason, the CA2 is able to follow spiking activity for very long periods of time. Yeah, in terms of the plasticity, they might have, it might be facilitated by the high calcium buffer that they sustain. But yeah, it's a good point, yeah.
Thanks.
So, as to ask Cliff's question from the previous talk, so you see these shockwave ripples also during awake states, not just during sleep. So do you see the barrages as well? Are they always in this nice sequence?
Yeah, we are looking into that. We might see them during awake as well, but they are not as frequent, yeah. Which is the same for shockwave ripples, yeah.
Other questions? Okay, if not, then let's thank the speaker again. Thank you very much.